This is a diagram out of a recent review paper by Karen Cassiotti. If you're interested in the nitrogen cycle, particularly in terms of the dissolved species and the processes that control it, this is a wonderful paper, wonderful review paper. And this really summarizes sort of the direction of change in the nitrogen isotopic composition of different species associated with different processes. And most of them, we're looking at a residual reactant, or in some cases a product. You know, here, nitrogen fixation decreases the nitrogen isotopic composition of particulate organic nitrogen. Assimilation or ammonification, so loss of particles, increases the nitrogen isotopic composition of particulate nitrogen. Ammonia oxidation, the residual reactant, ammonium in this case, will increase. Nitrite oxidation is the odd one. That's that inverse kinetic isotope fractionation that results in a decrease in the isotopic composition of nitrite, both for nitrogen and oxygen. Denitrification results in a residual reaction in which nitrate and oxygen isotopic composition of nitrate increases. But you can see that here. The net effect of these processes is described nicely in this figure, where we have the delta 15 N value of nitrate. Why nitrate? Nitrate is the main form of nitrogen in the ocean. And whether or not it adds nitrogen or takes nitrogen away, so source versus sink. Let's look at newly fixed nitrogen. That's adding nitrogen, so nitrate concentrations will increase and the isotopic composition changes a little bit. Sedimentary denitrification takes nitrogen away, but because it happens in a closed system, there is effectively no effect, there's no influence on the delta 15 N value of nitrate. Nitrate assimilation, again, removes it with a small isotope fractionation, so it will increase the nitrogen isotopic composition of the residual nitrate. And water column denitrification can change the nitrogen isotopic composition of nitrate a lot. Okay, it turns out that those high concentrations in the deep ocean, anywhere in the ocean, if you measure the nitrogen isotopic composition, either in the Atlantic, the Indian Ocean, or the Pacific, the nitrogen isotopic global average is about 5 per mil. So you will find nitrate in the deep Atlantic, the deep Pacific, the deep Indian Ocean has an isotopic composition of about 5 per mil. So what it means is that these processes of adding and subtracting are pretty well balanced, and it maintains that, that isotopic composition of about 5 per mil. And the concentration doesn't change a whole lot either. You know, it's a fundamental question. If the major form of nitrogen in the ocean is N2 gas, why is the isotopic composition of nitrate not 0 per mil? You know? Well, it's because of processes like denitrification, both sedimentary and water column denitrification, nitrate uptake versus nitrogen fixation, that drive that. And it's a balance between the isotope fractionation associated with inputs and losses. Okay, we can recognize some of these processes in different regions of the ocean by looking at profiles of nitrate concentration and nitrogen isotopic composition with depth. And that's what I've listed here. Here's depth and the delta 15 N value of nitrate in the eastern tropical North Pacific, the Southern Ocean, and the North Atlantic. The eastern tropical Pacific is one of these regions where it's relatively sluggish circulation and high production. So there's low oxygen. It's one of these oxygen deficient regions here in midwaters. And this is the nitrate concentration. It's barely measurable in the surface, but it increases. And then there's these little weird inflection points. These inflection where it's not following a nice smooth curve is because of denitrification. Let's follow the nitrogen isotopic composition. It kind of goes down here, and it goes way up, this increase in the nitrate isotopic nitrogen isotopic composition is due to, you break NO bonds. And it's a kinetic isotope effect. Bonds to lighter isotopes are weaker than bonds to heavier isotopes, so you lose light nitrogen, and the residual reactant increases. So this increase in the nitrogen isotopic composition with depth is indicative of denitrification. If we look at the nitrogen isotopic composition of N2 in a region like that, we see that the delta 15N value of nitrate increases here from around, it looks like about 7, 8 per mil to 15, 16 per mil, and then decreases again. Commensurate with that, the delta 15N value of N2 gas decreases by about, maybe half a per mil. Why is there such a small decrease in the product of this reaction? There's a, 7 to 16 per mil here. That's almost 10 per mil, but here half a per mil. Why is that?
Because more of them pushed in the atmosphere.
Yeah, isotope mass balance. Yeah, exactly. So you wouldn't expect to see a big signal there. So that's a hard measurement to make and to see differences because the background is just so high. So this is just isotope mass balance. We're adding a small amount to a big pool, but it's isotopically different, and we see exactly the pattern we would anticipate from that reaction. Okay, so the Southern Ocean is one of these high nutrient, low chlorophyll regions. Most areas of the ocean, the isotopic composition of nitrate can't even be measured in the surface ocean because it's gone. The isotopic composition of particles then reflects that of the upwelled nitrate from deep water. So it converges on about +5 per mil. But in regions where there is excess nitrate, and here we're looking at concentrations that are, 25 to maybe 35 micromole per liter. The isotopic composition here, look at that, it increases within the euphotic zone. Again, there's uptake of nitrate. The light isotope is preferred, so we're looking at the residual reactant that shows an increase. So this is indicative of assimilation. The North Atlantic is, again, one of these oligotrophic regions, so nitrate assimilation. We have nitrogen fixation here. Nitrogen fixation, we're seeing it in nitrate. Nitrogen from the atmosphere dissolves in the ocean. It's fixed by diazotrophs. And then there's recycling that occurs. So that light nitrogen associated with N2 fixation is released. And that's what we're seeing here. Low values, here's 2 per mil, and it's of nitrate. So this nitrate is a mixture of nitrate, the isotopic composition of nitrate that's fluxing from deep water, as well as the inputs of nitrogen fixation. The nitrogen is fixed. It's assimilated, but then there's ammonification that occurs and nitrification where it ends up in nitrate. So the nitrate-nitrogen isotopic composition shows this decrease and then an increase with depth to this roughly 5 per mil. Where's the 5 per mil here? In the deep ocean. Okay, so those are just profiles of regions that are dominated by different mechanisms of nitrate uptake and loss. Okay, so in the Southern Ocean, this is a region which shows an association of nitrate assimilation. Let's look in detail at how that occurs. Again, I don't want to belabor the point, but flux in, flux out, and an isotope effect associated with fixation of that. In this case, we have nitrate. Nitrate is transported into the cell. There's a diffusive boundary layer, and then there's uptake at the cell surface, which is an active transport. We don't know what the isotope effect associated with that transport or the diffusion is. It's probably small. I think Danny Sigmund's laboratory at Princeton University is working on measuring that. But nitrate gets in. The nitrogen isotopic composition probably changes a little bit in its journey into the cell, but not a lot, like carbon, about 1 per mil. Then there's nitrate reduction. So it's nitrate reductase. It's the same enzyme that's used by bacteria. The isotope effect is somewhere between 50 and 30 per mil. It's reduced to nitrite. Nitrite typically doesn't accumulate in the cells. So it doesn't accumulate in the cells. So really the last step in terms of fractionation between nitrate external to the cell and that fixed in organic nitrogen is probably nitrate reduction. Nitrite is reduced to within the chloroplast to ammonia, and that ammonia goes into organic matter. So really we're talking about the flux of nitrate in and its reduction and the flux out. And of course we do need efflux out of the cell to have any fractionation. Okay, so if we look at the Southern Ocean and the nitrogen isotopic composition of nitrate as a function of depth, we can treat it as a closed system. So we can look at basically a Rayleigh distillation, the delta 15N value of nitrate versus the log of nitrate concentration. What we see is that it falls on a nice line with a slope of around 5 per mil. So that is the isotope effect associated, describes the isotope effect associated with nitrogen assimilation. Okay, we can also study this. You know, almost anywhere in the ocean that you look and you can make this measurement where nitrate is not completely utilized by the cells shows this kind of relationship. You can look at any high nutrient, low chlorophyll region, the subarctic Pacific, equatorial Pacific, the Southern Ocean, regions that have excess nitrate available, we see a relationship like this. A pretty constant fractionation associated with nitrate assimilation.
Are you going to see, are you going to see this in human systems too, where they're putting in excess inputs of nitrogen?
The question is, are we going to see this in human systems or regions?
By human nitrogen inputs?
Yes. Yes, we will see the same fractionation associated with nitrate assimilation regardless where that nitrogen comes from. So you begin to think about that in terms of a region where there's nitrogen fixation that's dominating, where nitrogen isotopic composition in the surface water is around 2 per mil. You know, if there's not complete utilization, you would expect particulate nitrogen formed by phytoplankton to be about 5 per mil, less than that. Okay, makes sense. But in regions where there's denitrification that has nitrate that's being supplied to the surface ocean with a delta 15N value of plus 20 per mil or plus 15 per mil, we're going to see a different absolute value, but the fractionation will still be the same. And even in regions where there's input of anthropogenic nitrogen.